In this mysterious solution extravaganza, we discovered the concentrations of various substances based on our knowledge of osmosis as AP biology students.
Using disguised substances, we designed an experiment to identify the sucrose concentrations of each colored solution. Based on our knowledge of hypotonic, hypertonic, and isotonic solutions relative to plant cells, our goal was to classify the concentration of each of the substances. The data collected from this lab allowed us to order the solutions according to their estimated water potential. If we place the 2 by 2 centimeter potato cubes in different concentrated solutions of 200 milliliters, then we will be able to create an order of highest concentrations to lowest concentrations based on the difference in cell mass and appearance. If the solution has a low concentration, we expect the cell to swell. If the solution has a high concentration, we expect the cell to shrink.
In order to conduct the experiment, we used paper towels, russet potatoes, beakers, thermometers, a knife, a balance, a ruler, petri dishes, tweezers, the water solutions, parafilm, and a tray. In order to conduct the experiment, we collected all the materials. Next, we cut 6 2 by 2 centimeter potato cubes. Then we filled each beaker with 200 milliliters of a substance, and one with water as the control. We took a picture of each cube and assigned them each a color. We weighed each cube before merging them into a substance, and created a table with this info. We covered each beaker with parafilm and let it sit overnight. After letting the beakers sit overnight, we weighed them all again. We were hoping to compare the previous weight with the new weight the next day. We took more pictures and more visual observations were taken as well.
When the potatoes had been placed in concentrated solutions overnight, all potatoes had gained weight. Each potato had expanded, all while at a temperature of about 21 degrees Celsius. After returning the next day, we found that the cube of the potato in the water had increased by 199%. That's how much the mass increased. The cube of the potato in the yellow solution had its mass increased by 192%. The cube of the potato in the purple solution had its mass increased by 206%. The cube of the potato in the blue solution had its mass increased by 203%. The cube of the potato in the green solution had its mass increased by 173%. And last, the cube of the potato in the red solution had its mass increased by 219%.
Since we got some weird results, we decided to interview another group to compare.
Hypothesis.
Our hypothesis was that the potatoes would shrink when they were in an environment that was more hypertonic, and they would swell if they were in an environment that was more hypotonic.
Okay. So how were you gonna measure this in your lab?
So we measured it by weighing the potatoes before the experiment and then weighing them again afterwards.
And what was the purpose of weighing them?
To show the difference of water concentration in the potato cells.
Okay. So what were your variables?
Our independent variable was the type of solution we put the potatoes in, and the dependent variable was the change in weight after they were placed in the solution.
And did you use water as a control?
We actually use the size of the potato as a control.
Okay. So was the size of your potato a constant?
It was.
And how did you cut your potato?
We used the cork borers, so they were all the same width and same size.
Okay. And were they the same weight? Like, did you measure the weight in addition to
They were
cutting them the same?
approximately the same. They differed a little bit, but we just compared the weights of the same potato before and afterwards.
The differential.
Right.
And so, how much of each solution did you use?
We used about 50 milliliters.
Okay. And so, what did you end up finding out? Like, was your hypothesis correct?
Our hypothesis was correct because the potatoes that had gained mass were in the hypotonic solution, and the potatoes that lost mass were in the hypertonic solution.
Okay. So, did you have any like major sources of error?
Some sources of error, our measurements of the water that we put the potatoes in weren't exact, and also the original sizes weren't exact for each potato.
All right. Thank you.
You're welcome.
Although our results were thrown off by a source of error, I will discuss the inaccurate results we found. Through this experiment, we should have observed different changes in mass with each of our potatoes. However, instead of having a variety of mass changes, all of our potatoes increased in mass. This would allude to the idea that all the solutions resulted as hypotonic. We knew this wasn't true because the whole nature of the experiment was to record different changes in mass and observe all the following solutions: hypotonic, hypertonic, and isotonic. As a whole, our group was not able to successfully match all the glucose solutions. Instead of having a variety of solutions, some hypotonic, hypertonic, and isotonic, our results concluded that all the solutions began as hypertonic and resulted in hypotonic cells. Our main source of error resulted from a low battery percentage in the scale we used to measure the initial mass of the potatoes. Due to this unforeseen error, we were forced to use a different scale for the remainder of our experiment. Therefore, the scale is no longer a controlled variable in our experiment, and since our results were decidedly wacky, the most reasonable conclusion would be that the scale change resulted in unclear and unexpected data that negatively impacted our results. Of course, other experimental errors could be possible, such as the freshies messing around with their graduated cylinders, or the potatoes not being perfectly symmetrical. However, the scale change was by far the most noticeable error, therefore being the most prominent source of error in our experiment. In reference to the cell situations, any cell that had lost mass would have began as hypotonic because the water would have been driven to move outside the cell, from a higher concentration gradient to a lower one. Cells that gained mass would have begun as hypertonic because over time osmosis would have prompted the water to move inside of the cell. All of our solutions would have been considered hypotonic, since all the cell's mass increased. However, we know that this is inaccurate. Any cell that experienced the least amount of change would have been considered isotonic, but as aforementioned, none of our cells displayed this. The water potential of a potato cell can be calculated using the formula negative ICRT. In this case, the sucrose has an ionization of 1. The molar concentration would be 0.2, or 2/10, based off the other group's accurate data. The pressure constant would still be 0.0831, and the temperature was originally calculated as 21.4 degrees Celsius, or 294.4 degrees Kelvin after it was converted. By plugging all these numbers together into the equation, we can predict that the water potential of a potato cell is approximately negative 4.89.